Swayam Prabha. Digital India. Educated India.
So, we will start where we left off the last day. We were discussing the implication of kinetics, right, in protein folding. So, the topic we are going to start off with today is known as phi value analysis. Now, you will see it is an exquisite way of trying to determine how the transition state is affected, because the only way the transition state can be probed is by kinetics. Right. There is in protein folding, at least it is very hard otherwise to figure out what is happening in the transition state. So, what is this phi value analysis? So, I will go in more detail. Let us look at this formula first. So, what it says is phi unfolding, that means you are unfolding a protein, you are starting from the folded state, you are going to the unfolded state. What is it equal to? It is equal to delta delta G dagger minus F. That means, because you are going from the folded state to the unfolded state, right, it is phi unfolding, right. Hence, the difference between the transition state and the folded state is the one which is your energetic barrier, and you are considering a delta delta. Why it is a delta delta? Remember, it is a delta delta because if you are taking just one protein which is the wild type, then it is delta G dagger minus F. But now you are comparing two proteins. What are the two proteins? In one protein is the wild type, that means you have not made any changes in the amino acid sequence. In the other protein, what you have done is you have made a mutation, and hence the difference between these two delta G's is referred to as delta delta G. Okay. So, the top one is the one which is related to the transition state, as you can understand. What is the bottom one? The bottom one is the one which is related to what? Tell me in the simplest term. It is related to an equilibrium term. So, it is essentially the difference between the unfolded and the folded state between the two proteins, one being the wild type, the other one being the mutant. And to make it a little more clear for you, this is how it goes. So, that means phi unfolding is equal to the change in the barrier minus the change in the folded state, which is in the top one, the numerator, and the denominator essentially is delta G u minus delta G f. Okay. Again, this one comes from equilibrium thermodynamics, and this one we get from kinetics. Okay. See, the bottom one we have already referred to before. We can always have equilibrium free energy changes for proteins. Remember all those transitions we talked about before, right? That is easy for us to have, but for having the same information with respect to the transition state, we got to go through kinetics. Now, why we are going through all this, or why was this idea developed? Now, this is why it was developed. Remember, we started this when we talked about before actually coming to this, the topic we started on is or was rather the nucleation condensation model, wasn't it? It was a nucleation condensation model. And if you would try to figure out, which amino acid is being involved in the transition state towards the final structure formation, this is something you would be doing. Again, what it means is, what you are trying to figure out now is, if the protein has 150 amino acids, right, not all amino acid would equally affect the folding of the protein. Instead, what you would do is, say a protein forms a hydrophobic core, right, which is the interior of the protein, and you would know for the protein to be stable, the hydrophobic core, the interactions have to be pretty well developed. If that is so, then what it would also tell you is that those amino acids in the hydrophobic core would be one of the key components for the protein to remain folded, right. If that is so, what you would do is, you would try to target one of those amino acids, replace it with another mutant, and see to what extent it is affecting your folding or unfolding. That is essentially what you will be doing, okay? And that is essentially what you are trying to figure out. And to end this part of the discussion, this tells you the residue level. It is like a residue level interrogation of how a protein folds. That means, you pick amino acid by amino acid, you change that amino acid with another amino acid. I will tell you under what conditions or what are the approximations you can do, or what changes you can do. If you would change this amino acid with another one, then you would absolutely be able to figure out to what extent this amino acid was being involved in the folding or unfolding pathway, and this way you can pick out a lot of amino acids. That is essentially what you want to do, because getting residue level information in proteins, especially for a two state folding protein where there are only two states, folded and unfolded, is very hard. Is it clear? So, again we have to get residue level information. Now, let me just take you back to something you would know. Remember, if you would try to figure out whether that hydrogen is involved in a certain reaction, right, whether that bond involving the hydrogen is involved in a certain reaction, what would you do? You replace it by deuterium. Remember this primary kinetic isotope effect, secondary kinetic isotope effect. What are you trying to do? You are trying to figure out whether that bond is actually involved in the reaction, and then to what extent is it involved, right? Exactly the same thing you are doing here, right? Instead of a bond, what are you replacing? You are replacing an amino acid. Okay? That is essentially what it is in a nutshell. Okay? Then what is your strategy? Your strategy is this. The first one is, you have to make mutants in such regions of the protein that contribute to the stability. Now, it makes sense, right? If you take a certain mutant which has very little effect on the stability of the protein, you are not going to get much information, because you know finally the final structure of the protein or the native structure of the protein is not exactly determined by that amino acid. Hence, the change whatever you would be getting would be either very small or insignificant. So, you would have to, by logical choice, look at a certain region of the protein which is inherently involved or intrinsically involved or very importantly involved in terms of its interactions and with its neighbors and all these things. So, you pick out the hydrophobic core, that is one of the things which we always do. Okay. Then now, each mutant is used as a structural probe during folding and unfolding of the protein, that is what I said. That means, if you would be doing the kinetics, I will show you later, you can look at the folding arm or the unfolding arm, and you will be able to figure out to what extent this amino acid is affecting this arm or that arm. Okay. Now, wait for that one. Now, what you would do is, you would do kinetic measurements, right? You would do kinetic measurements on both the wild type and the mutant. So, remember, if you go back to the phi f, the phi unfolding rather. So, phi unfolding was equal to what? Delta delta G hash minus f, right? So, that means, if you would do kinetic measurements on the two mutants, what would you get? You would get this numerator. The denominator is coming from what? Equilibrium measurements. For that, you do not have to do kinetics, because that is essentially equilibrium, and hence you compare those. So, then that is what the last statement says. The above is correlated with the change in the equilibrium free energy of unfolding, because I am talking about phi unfolding. Parallelly, I can also talk about the reverse process, that is phi folding. Here I am starting from the unfolded state going to the folded state going to the unfolded state. I can also start from the unfolded state and go to the folded state. Either way, no problem at all. Okay. So, let us look at a free energy diagram, right, and let us try to keep it very simple. We are talking about a 2-state protein, right. So, let this be the wild type, say this is the folded state, because we were talking about unfolding. So, we start from the folded, go to the unfolded. So, this is the transition state, right, and say this is your unfolded state. Okay. So, wherever you have these solid lines, this one belongs to the wild type. That means, you have not done any changes in the protein whatsoever. It is how it is found in nature, that is it. So, if I just join these, say this one right. So, this is the transition state of the wild type. Now, what you do is you do a certain mutation, right, and say something like this happens. Now, this is just a profile of what changes we are going to look at. Okay. So, all the dotted lines are your mutant. Okay. All the dotted lines are your mutant. So, here also I will be having a similar thing. Okay. This is my free energy, this is my free energy in the y axis, and the x axis have a certain reaction coordinate, it does not matter what it is. Okay. Now, what is this one? So, from here to here, from here to here, what is this one? If this is a transition state, right. So, this is the transition state for the wild type, this is the transition state for the mutant. So, then this one I can write as G dagger minus G F. Okay. Good. Now, if this is the mutant for me, right. So, this dotted line is a mutant. So, then for the dotted line I can write this is what is G prime, the prime refers to the mutant, minus G, this is F. Is this clear? So, one is the free energy difference between the transition state and the folded state of the protein in the wild type, which is G hash minus G F. The other one is for the mutant, which you see on the left hand side. Okay. So, now, because you see the folded state has also moved and the unfolded state has also moved with respect to the wild type for the mutant, this one I can say this is delta G F, okay, and this one I can say it is delta G U, right. Can you tell me what this one would be now? This difference between these two, what should I write? So, this one is delta delta G, is not it, right, because I am going from the folded state to the unfolded state, I am looking at unfolding. So, the idea of this figure is, just look at what this figure is trying to tell you or portray to you is essentially these things. What are the thermodynamic parameters that are involved in this actual transition? So, that means if the mutant is changing my folded and unfolded state, then the change for the folded state would be given by delta Gf. The change for the unfolded state is given by delta Gu. G hash minus Gf is the Ts barrier. G hash minus Gf with the primes is the same barrier for the mutant. So, if I take a difference between these two barriers, I will be getting delta delta G. That means the difference in barriers of the two transition states, which is delta delta G hash F. Okay. Is it clear what we are talking about now? So, this is what we are going to talk about throughout in the rest of the class. So, now you can understand one thing, if instead of going from the folded to the unfolded state, if you would go the other way around, what would happen? This delta Gu and delta Gf would remain the same, no problem, but what would change is, instead of this minus F, what should we write? Minus U. That is the only small change that would happen. Clear? So, if from left to right I am talking about phi unfolding, then from right to left I should be talking about phi folding. And what will happen in my phi unfolding would be that wherever I have F, it would be replaced by U. Okay. I will come to that again later. But anyway, is this diagram okay for you, at least to start with? Okay. Now let us actually talk about this phi unfolding, this phi value. See, if it is a ratio, typically what would we think? It would lie between 0 to 1, right? We are talking about a ratio. Now, that is what it is essentially. So, then the two extreme cases are, one is phi unfolding 0, the other one is phi unfolding 1, right. So, let us look at those cases in a stepwise manner. So, suppose I have phi unfolding equal to 1. If I have phi unfolding equal to 1, now from before, which is the first slide, what was phi unfolding equal to? Phi unfolding was equal to, if you look at the second equation, it was delta G hash minus delta G F, right. Then what was the next one? Delta G U minus delta G F, because I am going from the folded to the unfolded state. U is my final, F is my initial, right. Now, if phi unfolding is equal to 1, so if this is equal to 1, that means what can I write? If so, now phi unfolding is equal to 1 for me. So, I can write delta G U minus delta G F is equal to delta G hash minus delta G F, is not it? Very simple, because phi unfolding is equal to 1. If that is the case, what am I left with? Delta G hash is equal to what? Delta G U. Now, try to understand the significance of this. What are you saying? What are you saying is, if my phi unfolding is equal to 1, then whatever change I have in the transition state is equal to the change in free energy of the unfolded state. Now, that is what you would expect, right. Phi unfolding is equal to 1, that is what it means. To put it in words, what it means is, whatever structure I am looking at, the protein is as unfolded in the transition state as it is in the unfolded state for both the wild type and the mutant. Do you understand this or not? That means there is a 1:1 correspondence between what? The change in free energy of the unfolded state and then change in free energy of the transition state for both the mutants. That means this I do for the wild type and then I do it for the mutant. Is it clear? So, that means I can say based on this, the Ts, which is the transition state, is as unfolded as the unfolded state for both the wild type and the mutant proteins. Huh? Say that again, I didn't understand. So, what we are saying is that the transition state is as unfolded as the unfolded state. So, let me write this unfolded state which is U for both the wild type and the mutant proteins. That means, whatever change in unfolding free energy you have in the unfolded state would be exactly reflected in the transition state. Is it clear? Okay. Good. Now, if this is the case, what is the other case? The other case would be phi unfolding equal to 0. If phi unfolding is equal to 0, what does it mean? If phi unfolding is equal to 0, then based on this equation, if phi unfolding is equal to 0, the numerator is 0, then I can write delta G hash minus delta G f is equal to 0, or delta G is equal to delta G f. Now, what is the difference between phi unfolding equal to 1? The phi unfolding equal to 1 says whatever change I have in the unfolded state gets absolutely reflected in the transition state. What does this one say? What this one says is these two are not related. That means, if it is a two state situation, we are not talking about the unfolded state, then we are talking about which state? The folded state, because it is a two state scenario, it is always a two state situation. So, then what we can write here is the transition state of the protein is as folded, the transition state of the protein is as folded as it is in the native or folded state F for both the wild type and the mutant proteins. Okay. Tell me, is the difference between these two clear to you? If phi unfolding is equal to 0, then you can see immediately what happens is my delta G, that is the change in transition state, is equal to what? The change of the folded state, right. So, now it is no longer related to the unfolded state, because whatever change in the transition state you are having, that is not getting reflected in the folded in the unfolded state. Where is it getting reflected? It is getting reflected in the folded state, right, and that is why this is clear, where delta G hash is equal to delta G f. Okay. So, this is one way of looking at it. That means, when you are moving from the folded state to the unfolded state, clear. Okay. Now, because we are doing this, let us do the other alternative. That means, let us talk about a folding scenario. If you are talking about a folding scenario, that means, you are moving from the unfolded to the folded state, then I will be talking about phi folding instead of unfolding, as simple as that, right. So, in this case, my phi folding would be equal to what now, tell me? What should I write in the numerator? Delta G, and I told you I am going from where? From the unfolded to the folded. So, this should be hash minus u, good. F gets replaced by u. You look at your previous equation, that was for unfolding. Now, you look at this equation, that is for folding, which you are going from the unfolded state. So, the energy barrier is in between what? The unfolded state and the transition state. Delta G, the initial it was u minus f, this one would be f minus u, good. So, if you write it again, phi f would be equal to then delta G minus delta G u over delta G F minus delta G U, right. This is a folding scenario, and if you take up any paper nowadays, people mostly talk about the phi folding. Okay. For a two state situation, protein, that means only denatured and the folded state or the native state, phi f is equal to 1 minus phi unfolding. This relation holds. So, phi f or phi folding is equal to 1 minus phi unfolding. See, it makes perfect sense. You think about this. If phi unfolding is equal to 0, what did you find? You found delta G hash was equal to delta G F, and based on this equation, what would phi folding be? 1. If phi folding is 1, what would you get? You would also get delta G hash is equal to delta G F, isn't it? And look at this. So, from this folding scenario, what are the two cases? So, that means if phi folding is equal to 1, if phi folding is equal to 1, I will not do the math anymore. If phi folding is equal to 1, what would you get? Tell me from here. The delta G U cancels out. So, what am I left with? Delta G hash would be equal to delta G F, and this was equivalent to what? Phi unfolding equal to 0. That is what we found, right. If phi folding is equal to 0, what would that imply? From here, delta G hash would be equal to delta G U, good. Delta G hash would be equal to delta G U, and this is something where we saw where phi unfolding is equal to 1. Clear? So, these can be used interchangeably, no problem at all, provided two state, absolutely, and there are also certain cautions or assumptions you have to maintain when you do these mutations. Okay, it's just not straightforward when you do these mutations, okay, because you are changing a lot of parameters out there. Okay. So, let us look at these assumptions and then I will give you a practical example. What are the assumptions of the method? See, mutation should not alter pathway of folding. Now, this is logical, right. If I have a certain amino acid A1 and I change it to A2, and the way the protein was folding initially, the pathway changes, then we cannot compare anymore, because now we are comparing two different pathways. It is not a fair comparison, right. So, one of the main assumptions is that this one is not changed. So, you can understand one thing, this imposes a huge restriction. The restriction is you just cannot change any amino acid arbitrarily with any other amino acid. It has to be a logical choice. I will tell you what those choices are. Okay. That means the total energetic change would not be that huge, otherwise there might be a drastic change in the protein folding pathway as such. Mutation does not significantly change the structure of the folded state, right. Now, this is also logical. If my folded state structure is fully changing, then possibly I am comparing two different structures in the transition states, then again that is not a fair comparison. Okay. Same, mutation does not perturb the structure of the unfolded state too much. Okay. So, remember one thing, unfolded state anyway does not have too much structures to start with, not like the folded state as such. But this does not mean that if you are doing mutations, it would not affect the free energy of the unfolded state. It would. So, that is something you also have to keep in mind. And last but not the least, there are also some other ones, but we will not be talking about those. Those mutations should be avoided which add new functional groups to make extraneous interactions within the protein not found in the wild type. That means, you cannot make a mutation, because say suppose there was an amino acid to start with, you replace that amino acid and the new amino acid say forms a salt bridge. So, there is a huge change. The moment you have this extraneous interaction, also new hydrogen bonding coming in, you cannot use this, because then again you have to take care of all those interaction energies. How would you do that? Transition state comparison has to be done with minimal structural change and minimum change in what? The type of the folded structure and all those things. Also, it should not introduce stereochemical clashes or a totally a new stereochemical disposition. Otherwise, it might destabilize or change your folded state unfolded state to a huge extent. So, these are the assumptions, right. So, now you can understand how hard it is to do a 5 value analysis, because the simple thing is, when you would pick out an amino acid, that amino acid will be interacting with a lot many other amino acids depending upon which portion you pick out, right. And no matter how cautious you are, there would be certain difference in changes. That is why it is very hard for you to find a 5 folding which is equal to 1. Why? Because intrinsically you would always be making some unwanted changes no matter how careful you have been, and this you are talking about a change which is at a very important part of the protein in terms of the stability of the protein. That is why it is very hard to find, very hard, but you will be finding high phi values, okay, which are phi f which are close to 1. So, then what are the, so these changes are referred to as non-disruptive deletions, a logical word, because you are not trying to disrupt the structure, or you are trying to keep the disruption to the minimum. So, what are the changes? The first one, you can go from isoleucine to valine to alanine to glycine, right. Even in this you would be seeing that there is a pretty much change in the size, but still if you talk about this delta G cavity and all these things, they can be taken care of, and people have done that. The next one is leucine to alanine to glycine. The third one is styrene to serine. The fourth one is phenylalanine to alanine to glycine. So, if phenylalanine to alanine is actually a drastic change, but people still have been able to do that with a certain amount of confidence. Okay. Now, I would just ask you to focus on this mutation, alanine to glycine. If you would ever look up these 5 value analysis or mutation analysis, then what people do is the people do something known as, based on this, alanine to glycine scanning. See, what does it mean? See, it is not easy to do mutations in all proteins, but alanine to glycine is still a mild mutation. So, typically think about helices, where helices are pretty well packed. You cannot just change an amino acid arbitrarily, but if you are trying to find out, to what extent that part is involved in the transition state or not, what you do is you take alanine and mutate it to glycine. That is what they say by an alanine to glycine scanning. That means you scan along the amino acid chain where you replace alanine by glycine and look at the corresponding changes in your phi values. Okay, this is something which has been routinely used, especially for proteins where it is very hard to do mutations. Okay. Now, since we are talking about non-disruptive deletions, let us look at a disruptive deletion. So, you avoid phenylalanine to leucine, because this brings about a huge change in stereochemistry. So, this is something you will have to avoid, okay, based on whatever assumptions we have taken, right. So, just keep these in mind. I cannot go through a lot of examples, but at least I will show you one or two, so that I can put forward the idea or make it a little more clear, right. Now, you must be thinking about this. If we have been talking about phi f is equal to 0 and phi is equal to 1, what about the values in between? There is a fractional phi values. Now, these are very difficult to interpret. Fractional phi values can be interpreted in many different ways. Example, they can arise if a protein folds by parallel pathways in which parts of the protein are native like in the transition state of one pathway, but unfolded in the transition state of another pathway. So remember, so now it's not, you're not talking about one pathway, you're talking about parallel pathways. When you're doing this kinetics, you're looking at an average response. So in one parallel pathway it might be fully formed, in the other pathway it might not be formed at all. But when you look at it, what do you look at? You look at an average of these two. That means you look at a fractional value which is in between. In other words, remember we talked about parallel pathways when we were discussing protein folding funnels, right? So this many or oftentimes people take this fractional phi value as an evidence of parallel pathway existing in your protein folding or unfolding scenario. Okay. This is another case. If a side chain makes one set of interactions with one element of structure and another set with a different element, and these elements react in different ways during unfolding. Okay. Now, I cannot give you examples of both of these, but at least I will try to tell you something relevant to the first one. Okay, but just keep this in mind. Fractional phi values, that is why, are very difficult to interpret, and they make the analysis a lot more complicated. So that means you have to do a series of phi value changes, that means a series of mutations to figure out, is it exactly a parallel pathway or is it just a one pathway, I am having some other interactions coming in. For the protein discussion we started with, which was chymotrypsin inhibitor 2, what they have found is, by doing a series of mutations, that it is not a parallel pathway, but it is essentially one pathway. Okay. So that means just by doing one experiment of phi value, you cannot say whether it is a parallel pathway or a non-parallel pathway. That is essentially what I am trying to say. Okay. So look at a quick example of a fractional phi value. So this is what it is. In chymotrypsin inhibitor 2, there is an isoleucine at a 39 position, which is a part of the helix, and you are replacing it by valine, right. So this is located in the center of the alpha helix of chymotrypsin inhibitor 2. The side chain is in the center of the hydrophobic core and has many contacts with other side chains in the hydrophobic core. So immediately you understand, if you have to pick out a certain amino acid which is very essential for the stability of the protein, you would be picking out an amino acid like this, which has an important place in the structure of the amino acid in terms of the interactions it makes. Okay. Now, on doing this, what it was found was, there was a phi value of phi f of 0.5. See, what does it mean? If you think about this phi f of 1, it meant was that the delta G hash was equal to delta G f, right. Now, if it is 0.5, what does it mean? That means whatever you have in the native state, half of it is being developed in the transition state. That is what 0.5 means, right. So, whatever you have in the native state, half of it is developed in the transition state. That means there is a half resemblance, at least a 50% resemblance. Now, then how can we interpret this half? Now, think about this, how can you interpret this half? There are two ways you can do it. One is, either the isoleucine side chain could fully make some interactions, but have others completely unfound in the transition state. So, what does it mean? What it tells you is, I have done this mutation, right. Now, say I have 100 protein molecules. In the 100 protein molecules, 50 of the molecules have already formed that isoleucine, that part in the transition state, but 50 of the molecules have not reached the transition state even. But when I am looking at, what am I looking at? I am looking at an average. So, that means when I take the average, one is 1, one is 0, I get what? 0.5. Okay. Now, what is the other interpretation? This is what you can also say. What you can say is, okay, I would not go by that. Instead, I would say is, all the interactions have developed, but they have developed only to the extent of 50% as compared to the native state. That means all of those have developed, but only at the level of 50%. Do you understand the difference? In one case, the first one, what you had was 50% had fully developed, 50% no development of transition state. Now, in the second case, what you are saying is, see, I can also have a phi value of 0.5, which is midway, by having all of those developed, but not to the full extent of 1, but to the extent of midway, which is 0.5. So, if you are doing only one mutation analysis, how would you figure this out? It is very hard, because you already have 2 possibilities, right. So, then you will have to go on doing a series of mutations, but anyway the idea is not that, but the idea is to try to understand that having a phi value of 0.5 or having an intermediate phi value just complicates your data analysis, right. You have to do further experiments on that specific protein, on that specific, or doing some specific mutations. Okay. Okay, now let us look at this. So, this is now I was telling you, I will show you a free energy example. I am asking you about phi folding, that means I am going from the native state to the denatured state. Tell me what should the value of phi folding be in this case? Remember, if phi folding was equal to 1, what was the criterion? The delta G hash would be equal to what? The delta G F. Do you have it here? You do not have it, you see. This is essentially, what is this? This is essentially delta G, right. You see a change in the native state. Do you see a corresponding change here? There is absolutely no change. Instead, if you look at this here, if you look at this and if you look at this, these are very similar. That means, I can say in this case that my delta G hash is equal to delta G u, is not it? I can say delta G hash is equal to delta G u. Why? Because this change, whatever change you have here, this change not reflected in the transition state. So, that means what should be my phi f equal to 0 or phi u is equal to 1. Clear? This is how your transition state would look in terms of the changes in free energy. So, then now you can realize, if this is phi f is equal to 0, how would phi f equal to 1 look? Would it look like this? The same amount by which my native state is perturbed, my transition state is also perturbed by the same extent. See, what does it mean? It means is if I am doing a mutation and if my native state is getting perturbed, or if my transition state is getting perturbed very similar to the native state, that means that portion is being formed in the transition state before going to the native state. I am repeating, suppose you have a certain portion of the structure, say hydrophobic core, formed in the native state, you know there is a native state. You do a mutation, you saw that the free energy change, there is a certain free energy change in the native state, there is exactly a similar free energy change in the transition state. So, what does it mean? That means that hydrophobic core, which was formed in the native state, was actually being formed in the transition state, because both are being affected equally, right. So, that is why we said, remember when we were talking about this phi unfolding equal to 0 or phi folding equal to 1, what we said was it was as folded in this transition state as it was folded in the native state, as folded, that means to a similar extent. Now, if you try to rationalize phi unfolding, or rather phi folding is equal to 0, what would you say? If it is phi folding equal to 0, what you would say is that, which was the previous one, this native state, whatever difference you have, it is not reflected in the transition state. Then what would you say? What you would say is that whatever structural region I am looking at by mutation, that is not formed in the transition state, but is found in the native state. That means that portion in the transition state is still essentially as unstructured as what? The unfolded state. Guys, is it clear or not? I am trying to go at length, really slow, to try to emphasize how important this measurement is. It is not without flaws, let me tell you guys, it is not without flaws, but this is the closest thing you have. This is the closest thing you have to what? A single residual structural analysis, and that is why it is so important in the field of protein folding, especially when you are trying to reason out why an amino acid is there, or why did evolution actually try to figure out that sequence of amino acids in the hydrophobic core. That is always what a final goal is, try to reason out what evolution did. Clear? Okay. So, next day what I will do is, if I will start now I will not be able to finish. The next day what I will start with, I will start with something known as chevron plot, and a chevron plot, you will realize why it is so called. There is a V in the middle, and if you would look at this, I am looking at the slide again, you will see on the left hand side of the slide, there is a blue line, on the right hand side is a red line. These points are all experimental points. You look at the concentration of the denaturant, which is the concentration of guanidinium hydrochloride, which is the denaturant at the bottom. So, when guanidinium hydrochloride is low, your protein is in what? Folded state. When the guanidinium hydrochloride is high, your protein is in what? The unfolded state. So, now you can look at to the lower end of this, which is this region, to the lower end, this should be reflecting what? Rate constant for folding, because you are in the lower end of the denaturant. If you go to the higher end of the denaturant, what would it reflect? The rate constant for the unfolding situation, because your protein is already unfolded, right. And see what will happen is, this is the beauty of the chevron plot. Remember, when the protein is 50% unfolded, that means when you are at your Tm or Cm, rather let us talk about Cm, what happens to K equilibrium? 1. Okay. Now, if you are talking about folding or unfolding, does not matter, say you are talking about unfolding, right, then what will K equilibrium be? Equal to 1, right. That means that Ku by Ka for Kf by Ku should essentially be equal to 1. That means they should be essentially comparable. And see, you can easily figure out by doing a chevron plot, when they intersect, the point at the intersect means that Ku and Kf have the same value, and that is what your Cm. This is how you can also figure out what my Cm is and see whether it matches with your equilibrium studies. Okay. So, next class we will discuss this at length, and this would be the last in terms of what should I say, in terms of the fundamental aspect of of protein folding, or how to look at or how to study protein folding, because we have looked at thermodynamics. This is the kinetic part, and once we are done with chevron plot, what we would do is we would actually start looking at different ways of doing experiments, that means instrumentation. How do we do a kinetic analysis? That means CD, the probe, fluorescence and all these things. That is the rest of the course would be focused on that. Okay.